So the reason I'm speaking first is because Lorraine said she's very passionate about this and can speak for hours. So I realised if I didn't speak first, I wouldn't speak at all. And the other thing she kindly said was, David, you do the history and I'll talk about the future. So that told me a lot about my use-by date. So, but quite seriously, what I really want to kind of give you is a bit of background about how active the AGITG has been in this area and the reason why. So when I started getting involved in pancreas cancer about 1 million years ago, basically this was a cancer that was even more so, if it's possible, underserved and lacking in support than it is now, where things are really changing almost daily in news about possibilities, in the breadth of research, and finally support for research. It's still under-supported compared to our colleagues in many other areas, but nevertheless, it is improving, and so are the dividends that come, albeit incrementally. So the message I want to give you today is a bit about incremental change, about how things give you a bit of a timeline to show you that it does take time, and you have to have a lot of patience and a lot of patience who participate freely in the trials and research that change results, but patience to wait for not happening instantly and not going to be front page news the minute the trial is over. So when I started, one of the quotes that I saw early on in my career when I was starting to get interested in pancreas cancer was that the only hope, it's a terrible quote, that the only hope a patient with pancreas cancer has is the hope that the diagnosis is wrong. Now that's no longer the case. We now actually have things that we can do, and we do have evidence that outcomes are changing, albeit slowly. So the first trial that the AGITG was involved with was a trial called PA3, and that was in 2002. And what typifies all of those trials is that the AGITG has always punched above its weight. In other words, on a population basis, more of our patients have agreed to participate in the question and make a difference than any other country in the international studies we've been engaged with, and that's something to be proud of. And as a result, Australian investigators have got to get involved in innovative and groundbreaking studies early on because they recognise that both we and our patients are willing to participate in changing the nature of treatment. So the first trial back in 2002 was called PA3. And up until that time, there was only one drug that worked, called gemcitabine, and it was certainly not a cure, and it delayed at best by a few months the ultimate outcome. So a heap of trials had been done with adding something to gemcitabine, at least 20, nothing changed. Then along came this Canadian trial, and at the end, Lorraine's going to bookend it by talking about PA7, so things have changed, so pancreas. And they added a drug that was pretty sexy at the time, erlotinib, had changed the nature of treatment of lung cancer and was a very targeted drug, very specific, not like chemotherapy, a broad effect, affects all growing cells, only targeting a particular abnormality within a cancer cell. And this was the first trial that actually showed a difference adding an extra drug. It was a relatively modest difference, but something like overall about a 10% improvement in the 12-month survival, which was the first improvement ever seen. And it gave my colleagues around Australia some encouragement that a pancreas trial can be done. Up until then, a lot of my colleagues felt pancreas cancer trials were just too difficult, patients were too sick, it wouldn't be possible to do a trial. So it wasn't about the modest result, It was about the nature of the landscape suddenly being, yes, we can start to ask questions. Yes, we have patients who will participate, and we have doctors around the country who are willing to work together through the facility called the AGITG. So a little bit further on, let me get the timing right. Yeah, 2000, and also about a year later, we got involved in almost a bookend study. So this is about people in whom the cancer's already taken off, spread widely. It's not a curable setting. We try to improve quality of life and quantity of life, but not cure. SPAC said, well, for the 20% of patients who can be operated on, only 1 in 10 will be cured. Can we do any better if we give drugs afterwards? So they'd already done one trial and showed an improvement. So this was a confirmatory trial which compared the drug they'd already shown that had shifted 5-year survival. It had doubled, still very poor, from 10 to 20%, but a doubling in survival. What about if we used a more recent drug, gemcitabine, and we compared the two? And what they showed was that they got the same outcome. So it wasn't a fluke, the previous trial. It was a real change, and showed that this was a better tolerated drug to get the same bang for your buck with less toxicity for patients. So that actually was the first adjuvant trial ever done in Australia in pancreas cancer, and as a result changed practice, changed the PBS, and resulted in a practice change, and it's now a universal, adjuvant treatment is now universal in our country and funded as a result of the kind of momentum that the AGITG, by doing this trial, was able to achieve. Now, what I want to show you, which we weren't able to participate in, is the next SPAC trial, which just got released just last year, added a different drug, interestingly, an oral version of this old-fashioned drug called 5-FU, and putting them together, and got 28%. So from 2002 to 2016, we've gone from 10 to 30%. So that's reasonably significant progress.
And that's for patients who are...
The 20% who are operated on and given prevention chemotherapy for 6 months after the operation. Gone from, so when we look at that 7% result, if only 20% benefit and they improve by from 10 to 30, we're only going to move it to 9%. So you're going to look and say, well, that's not very good, but it's a very dramatic change over about 14 years. Patients and patients. That's what's required to go from there to there, and only a larger trials organisation like the AGITG can make those things happen and change practice. So we also decided to do some homegrown work. So we got involved in a trial of our own called Go For It Go, and the idea of that was to say, well, if we added radiation to chemotherapy in another 20% group, the group where it can't be operated on because it's too large to remove and it can't be done safely without and the patient surviving, but for some biological reason, and Lorraine's going to work out the reason and publish it next week, they just don't tend to metastasize. They just stick there and they keep growing. Eventually they metastasize, but could we make them live longer by adding radiation? And at the time, there was a lot of negativity about radiation because of a previous trial, but nevertheless, both patients and investigators around the country were willing to find out if it could be tweaked and done in a safer way with what we call conformal, which is very geometrically targeted ways of delivering the radiation to minimise damage to surrounding structures. And we were able to show exactly that. You could deliver it safely, you didn't end up causing major damage to the surrounding organs, and you therefore could potentially make a difference. This was a single-arm trial and it had a different approach. And then as a result, we were involved in a very large study called LAPO7, the largest study ever done in locally advanced disease, multinational. And the reason we were invited to participate, because the AGITG had a track record. And they said, we would like these people to partner with us because they have patients and doctors and nurses who want to do this kind of work and who'll do it well. And so LAPO7 was asking in a much larger group about, should you give chemotherapy and nothing else, or would adding radiation make a big difference? It turned out that it made a relatively modest difference. What it meant was that people who got radiation instead of just chemotherapy had a longer period of time without any treatment at all than the people who got continuous chemotherapy. So that was, again, what we're after is both looking at quality and looking at quantity and looking at the patient experience as a guide to what's useful as a treatment, not just a statistic of an extra 2 months of life. It's what does that 2 months of life mean to patients? And that's been a very fundamental theme, particularly in pancreas cancer, of the work that we do. And more recently, because of this work, a group of radiation specialists around the country competed for that $200,000 prize. We should have won it. I'm convinced we had the best offer, and we'll still be going out to get a grant, but using a very novel type of radiation called stereotactic, where you deliver a much higher dose, but in a very, very targeted way, so it doesn't, which it would otherwise do at that high dose, damage surrounding organs, but has potentially a much stronger killing effect to the tumour. So we hope that the AGITG will end up doing that study over the next few years, especially if there's more money coming into the Innovation Fund that we can apply for. So meanwhile, and Lorraine's getting worried that the reverse is about to happen, and she's right. You should go now, because there's no more time. I'm going to hand over to Lorraine, because meanwhile, a revolution led by two really, really determined people, a surgeon, Andrew Biancan, and a very, very bright molecular biologist, Sean Grimman, worked hard and got money from Cancer Council New South Wales, from the federal government and the state government, and they actually won the right to lead for the world what's called the Australian Pancreatic Genome Initiative, which Lorraine was a very important part of, and I won't take her thunder, she'll tell you about it. But the main message is that it's led to several trials. Lorraine's going to talk about one of them. And actually, I don't know that you were involved in PAN1, I've forgotten, were you?
Yeah, not very much.
Well, I might mention PAN1 then.
Yeah.
So PAN1 was kind of a precursor to this, but it was known, but only using retrospective analyses, that there might be a marker to help you choose between these two drugs so that you could get a better bang for your buck, give the right drug to the right patient. And Lorraine's going to show you how she's going to be doing that much better than that particular idea. And the idea was that we were going to randomise patients depending on the laboratory result that we got. The trial actually is, I guess, an example that there are pitfalls, and we don't always succeed in the trials that we do. And there were some technical difficulties getting results back in time. Lorraine will talk about that even more. That meant there were delays, and as a result, patients had to be treated before we knew the result. And eventually our funders felt that the trial was probably never going to recruit, and so they felt that they advised us that we should stop. Funnily enough, our recruitment took off just at the time they made that decision. So it's about patients as well as about patients. And it's about time that leads to change. But the lead time is long, it's not instant. So the last trial I guess I want to mention in that group is the LAPO7, I think probably that's enough. I might think of something, but I'm going to, yes, the GAP trials. Thanks very much, exactly. So what do we do next about this stuff? and it's the GAP trial. So together with, and that's the other part of the AGITG. I did that, the Australian arm of that adjuvant trial with a surgeon. It was only because the surgeon and I went around Australia convincing our respective colleagues, surgical and medical, that giving chemotherapy after a pretty tough operation could happen, that it ever happened, and that afterwards, when it was proven to be of benefit, that it was taken up by doctors and patients. So the GAP trial is a more recent one, asking the next idea, which is, if we give chemotherapy before rather than after surgery, will it be safe? Will it prevent anybody from being operated on? And will it show some encouraging results, as it has done in stomach cancer and some others, where neoadjuvant, before surgery, is becoming the standard of care? There's going to be probably a worldwide practice-changing trial. I'm predicting the result, we don't know it yet, but that's going to be led, that's being led from Australia in gastric cancer, asking exactly that, about radiation before rather than after, together with chemotherapy in gastric cancer. But that's for another lounge room day. We're only interested in pancreas cancer, the two of us. So neoadjuvant chemotherapy, could we convince surgeons that somebody who was operable should wait 3 months, 2 months of chemotherapy, a month of recovery, and will that be better than going straight to surgery? And the answer was that their fears were not realised, and the patients were brave to go into it. We did not impact on the ability to operate. There was no patient who ended up not going to theatre who should have gone to theatre. And some people were spared an operation that wasn't going to help them, was the other thing that we learnt. The time actually differentiated those who were going to get the most benefit from surgery versus those who would get no benefit. So the GAP trial, which hasn't been written up yet but will, completed 2 years ago. And some of my colleagues have said, why haven't you written it up yet? You know, we're all waiting, come on, we want the paper. We weren't able to write it because many more people were still alive at the predicted time to write the paper than had been planned for, and we've just had to wait because we need to know what the final survival is. That's a wonderful position to be in, and I'm extremely happy that I'm still waiting for us to get around to writing that paper. So 2002, 2017, changes in types of drugs, changes in the way we do things. And maybe the last thing I'll tell you that the AGITG has done is a consensus surgical conference that we held 2 years ago. There's been a lot of uncertainty about judging who's safe to operate on and who's not, and who's in this group we call borderline. If only it was just 20% smaller, well then we could operate. And what can we do to do that? So to do trials like that, you have to get definitions right. So we brought together all of the surgeons who are very focused on pancreatic biliary cancer and who do those kind of operations frequently, and got them in a room, and we worked out a consensus of how to classify who to operate on that was universally agreed on, and that we can now use in trials to be more accurate about who we give what sort of treatment to. So that's a nice lead into molecular accuracy, and Lorraine's going to talk a bit about that. Or any questions first about what I've covered?
You're talking about adjuvant treatments.
Yes.
What percentage of pancreatic cancer patients diagnosed at the moment would be regarded as operable?
So 20%, 1 in 5, are operable. The other 80% are either the 20% with the locally advanced disease, it hasn't spread but it's not operable. That definition is changing though because of this idea of borderline. So this 20% that we say locally advanced contains within it a group of people with the right treatment might be converted to operable. What we don't know, and that's the subject of some trials that we're even thinking about designing or perhaps joining in an international coalition, what we don't know is whether if you are locally advanced when you present and you shrink, does the biology change, or is it that operation or not, you'll still end up in the same place? And that's where, once again, segues nicely into molecular. Any other questions about old history before the new story comes your way?
When did they first start using gemcitabine for pancreatic?
Sorry, say that again?
When did they first start using gemcitabine for
So the first, which was the groundbreaking first drug that ever changed natural history, goes back to 1990 or so was the first study. Yeah.
Was it around earlier?
Oh yes, gemcitabine started with lung cancer.
Yeah, in the 70s.
But this was such a major watershed moment in the treatment of pancreas cancer because there had never been a drug before that had actually shown a difference in people being alive at 12 months. It was, again, with 5FU there was 1% alive at 12 months, and it was around about 20% alive at 12 months with gemcitabine. So it was a watershed moment in treatment.
Can I just ask one question, is that possible? I'm fascinated by gemcitabine. It sounds like a great name for a rock group out of the United States. But Abraxane was something that I had, and it actually helped in the whole process. I'm just wondering how Abraxane's gone in over the last...
So again, a nice timeline. So here we were, gemcitabine plus everything failed for 15 years. Except perhaps this drug, and only modest. So 2 trials really changed the landscape of pancreas cancer treatment. And the first was a trial that used 3 drugs that are quite popular in colon cancer, but had not been used in pancreas cancer except for 5FU. And that trial actually showed, compared to gemcitabine, 1 in 3 patients responding instead of 1 in 5. And a significant improvement in the number of people alive at 12 months compared to gemcitabine. First time the 2 drugs had ever worked, first time that there'd been a major change from 20 years of history. And then following soon after, a different combination, gemcitabine and Abraxane, with similar results, but less toxicity, more tolerability than that. And that tends to be used more for younger, fitter patients and those in whom you want a big shrinkage because that might change their operability. And gemcitabine and Abraxane perhaps more broadly applicable and with a similar improvement compared to gemcitabine alone. And that's opened the floodgates of trials using multiple drugs given together, which as recently as 5 years ago, people would look at me and say, 2 drugs is 1 drug too many, David, stop doing it. And that doesn't happen anymore. Everybody is 2 drugs unless there are a really good reason not to.
So thank you, and thank you all for coming. It's really nice to see you all here. I'd like to start by telling you how lucky I am because I've had some really lucky breaks in my career. And the first one was when I completed a science degree. I was able to do some molecular biology in 1988 at the Institute of Molecular Medicine in Oxford, and I learned how to do PCR, which was a very basic technique at the time, but took a lot of time, and since then things have taken off in molecular biology. Having done a science degree and worked overseas as a researcher, I came back to Australia and I did medicine. And during my medical degree, my mother died of cancer and I decided that's what I wanted to do. I wanted to do medicine and study cancer. But one of the main things I wanted to do was to be able to bring the laboratory research into cancer medicine. And so again, I was very lucky because I happened to be in the right place at the right time when the APGI started the very large genome project for pancreas cancer in Australia. So Australia was charged with sequencing 500 pancreas cancer genomes. And when we started this work in about 2009, there was an amazing uptake by the community. And I would say that one of the main things that gives us an advantage in Australia is that I know by first name all the people who work in pancreas cancer in this country. And we were able to get everyone together and to get tissue samples from patients who had had pancreatectomies. And we successfully sequenced more than 500 pancreas cancers and published their sequence data in a publicly available portal through the International Genome Consortium. And this has been an amazing achievement for a disease that had virtually no knowledge prior to that. However, it wasn't all easy. In fact, we were hoping to find some magic key to unlock the secret of this disease, but we didn't. In fact, the opposite is true. Having done the sequencing, we actually revealed more complexity than we ever expected. So the first part of the sequencing was doing exome sequencing, which essentially just sequences part of the genome, not all of it, and we were looking for mutations that might tell us what treatments we could use. The problem was that about 95% of patients had the same mutation, and then a further 50% had another mutation, and then it was just mutation soup after that. It was like there were 2 in this person, and then there were 3, and we couldn't work out any pattern. There really wasn't a pattern. There was a different set of mutations in every single patient. What that told us mostly was that pancreas cancer is a highly heterogeneous disease, and probably, for good reason, the same treatments don't work in the same way in everyone. And so what we were hoping to get from our work was, could we pick out some molecular targets that we could test? And so the APGI partnered with the AGITG in trying to do a molecularly guided clinical trial in pancreas cancer. It was actually very, very difficult. I think partly because we were ahead of our time. We started doing the trial, we designed the trial in about 2009. We started the trial in about 2011. But it was very difficult to do because our idea was, let's take the sequence data from people who we've sequenced on their primary pancreatic cancer, and if and when they develop metastatic disease, let's give them the targeted treatment based on that sequencing. The first problem was that most people had died by the time we got the sequence data. That was totally hopeless. However, that has changed a lot in recent years, and sequencing happens much faster. But at that time, in 2010, 2011, it was taking us many months to analyse all of the sequence data. And so many people died before we were able to change their treatment. So then we amended the study, and people were incredibly patient, and we went through a lot of amendments. We amended it, and we said, let's try and see if we can do this on people who present with advanced disease, and we'll do a biopsy, and we'll do the testing on that. We couldn't do full genome sequencing on those biopsy tests in a timely enough manner, but we decided to choose 3 low-hanging fruit, as you might say, and we just tested those 3 molecules for the RAS gene, for HER2 that's used in breast cancer, and for the BRCA-like mutations that people might inherit pancreas cancer from. Even just with those 3 low-hanging fruit, it was very difficult to find cases quickly enough to do the sequencing and to feed back that treatment information in time for the patient. Essentially, the trial failed.
So how long does it take to do the sequencing?
So nowadays, probably only about 4 weeks, but it's the analysis. But about the fastest is 4 weeks. Depends how much sequencing you do. So you can do targeted panels now quite quickly, but to do whole genome sequencing takes longer. And what we have seen from our work, and the wonderful thing about this work, is although that we didn't get instant results as we were hoping for, the amount of knowledge that we got is building and building, and we still have the best annotated clinical data along with that genomic sequence data for the whole world. Our data is better than everybody else's because we inputted every single detail about every patient, about what chemotherapy they received, how many cycles they received, et cetera, which many other units around the world were not able to do. So the resource that has been created is continually being used. People apply from all over the world, can we have a look at your data? We want to examine question A. We look at it, we have a committee that decides whether that's a useful idea, and then we mostly approve those applications. So it's a building resource. One of the really great things that came out of it was that actually on whole genome sequencing we were getting a lot more information, and we're able to isolate some people who have genomes in the pancreas tumour that are really very, very disordered. And we think that those people may respond to platinum-based chemotherapy, which is very important because, as David pointed out, right now, today, for first-line chemotherapy for advanced pancreas cancer, we have a choice between FOLFIRINOX and gem with Abraxane. If we could have some intelligent way of working out which one of those would be better for that person, then we would be ahead of things. So perhaps these genomes that are very, very disordered would probably do better with FOLFIRINOX first. Okay. We need to test these things in a prospective study, and I think that will come. We've sort of put our prospective studies of molecularly guided treatment on hold a little bit, I think, to wait for technology and analysis to catch up, for it to become faster, for it to become cheaper. And I think it will happen. So it has been very exciting, but I have to say that personally, I still feel like somewhat of a failure because I don't think we have changed very many people's outcomes using molecularly guided treatment yet. We have changed a few here and there, but not over a population, and that's what we really need to do. We need to make big changes, not little ones. And I say to people all the time, we're not going to give up until we make big changes, not little ones. Having said that, at the moment, I am amazed and excited by the end of nihilism in pancreas cancer, and we're getting new trials coming to us all the time. And one of the most exciting trials that's come to us recently is the PA7 trial that David mentioned, that's come from Canada, and that is a clinical trial in pancreas cancer using gem and Abraxane with immunotherapy. Now, many of you have heard of immunotherapy and all the wonderful things that immunotherapy is achieving in other cancers. In pancreas cancer to date, the results have been terrible. In fact, they've been very negative. It's probably done harm in trials so far. But this trial is different because it's going to be a trial of first-line treatment, and it's going to be in combination with chemotherapy. And again, I think something David mentioned, everybody's saying, oh, how can you do that to people with pancreas cancer? 2 chemotherapy agents and 2 immunotherapy agents. So it's going to be a 3-arm trial. In one of the arms, patients will receive 4 drugs together. And there's a lot of concern around the community as to whether that's a good thing to do. Will we hurt people? But I think we have to stop saying, as doctors, we have to stop saying we can't do that because it might hurt people. We have to start asking the person in front of us, would you be willing to do this study? I'll give you as much safety information as I can, but would you be willing to take the risk? And in my experience, most people are willing to take that risk, and I don't think we can put people off and say we're not going to do that trial because it's too hard. So we have to keep pushing the boundaries, and we have to also then take the responsibility for it if it is too hard, and we have to say that and publish it, and so that it doesn't hurt anybody else in the future. So there are really good reasons for doing it. The other, question.
Given that the journey with pancreatic cancer is often quite short, has the mutated cancer, does that suggest that the treatment at the end of that journey should be very different from the treatment at the beginning? Because is it a different enough cancer? And I suppose as an extension of that, you mentioned platinum-based drugs. Have you used oxyplatin or cisplatin?
Yeah, so I don't think, it probably doesn't matter which platinum drug is used, whether it's cisplatin or oxaliplatin, but generally speaking in pancreas cancer now, oxaliplatin is the treatment of choice. And probably the first treatment is the most important one, but you're right, what you do later is not really, you're not really given much information about that by the genomic analysis. You're given information about what to do first, but what to do later might be harder. There is one exception to that, and that is in people who have germline mutations in the BRCA1 or 2 genes. If they receive platinum-based therapy to start with, and that works, there is a case for then maintenance treatment with the PARP inhibitor olaparib. Now, there is a clinical trial around the country doing just that. So that's a very special trial, and I have to say I take my hat off to the company that's running that study because they are funding all of the testing for that study, which would be very expensive because the rate of germline BRCA mutation in our community is relatively low. So that's a very innovative and a very good study that's available here in Australia. So the other thing that makes me feel very hopeful is that for the first time ever, as far as I know. We now have clinical trials in the second and the third line for pancreas cancer. Now, in the past, we never thought that people would achieve a second-line treatment, and third-line treatment was unheard of. So I'm really proud to say that we now have a third-line trial available. It's a commercial trial, but it links with some really important work that is being done by our scientists here in Sydney. So there was a paper published very recently by Marina Pajic and Paul Timpson and their group about the molecular pathway involving involving CDK4 and 6. And there's CDK4/6 inhibitors on the market for use in breast cancer. And the clinical trial that we have available uses a drug like palbociclib in combination with some other new drugs for pancreas cancer. So that's really, really exciting. And we've never really seen that before. And it actually has some intelligence behind it, because in our genomics we did see a rate of changes in that CDK4/6 pathway. So that really makes sense. And I'm very excited to be able to do that too. So things are changing so quickly, as David said, almost daily. We're seeing changes, and the scientists in Australia really are absolutely amazing in this field. And the other thing that I feel we're getting better at is linking the scientists with the clinicians and trying to get those two things closer. The AGITG is absolutely essential for that process. We have, for the last 3 or 4 years, run a pancreas cancer research workshop where we're trying to do that, because until recently, I think people were working in their silos and not talking to each other, really facilitating collaboration between the laboratories and the clinicians, and having AGITG trials that are funded to fund these things that are not going to be done any other way, where we can look at molecular markers and try and look at some of these things that are being done in laboratories. Like Paul Timpson has some great work on metastasis, where he can actually look down a special microscope in life in a mouse and see cells moving from the primary out into the circulating system in an animal. We've trialed some drugs at stopping that process happening, and we can do it in animals. We need to do it in humans. So there are lots of examples of that sort of thing that it's going to come, and it's going to make such a big difference. So I'm very grateful to all of you, because you actually facilitate these changes. As we've said, it's not quick, but we're not going to give up. So thank you. I'm very happy to answer questions from any of you, or just talk to you about pancreas cancer. Have I missed anything, David?
No, but I would like to add something from a thread of Lorraine's. So the two of us represent quite an important example that actually isn't happening in every cancer. We both actually work in laboratories. We both have the pleasure of sitting with our laboratory colleagues, and we give them the clinical perspective. We tell them what we and our patients are concerned about, and they take our thought bubbles and turn them into something that can actually be done in a lab and in an animal that will provide an answer to help us design a trial. And that's actually, it's not unique, but it's actually quite a strong movement in Australia, and stronger in pancreas, to the best of my knowledge, than almost any other area. And it's not just the two of us. We have clinical colleagues in other places, particularly in Melbourne, who do the same thing. I go to a lab meeting every week, nearly every week, and sit, and they present data, and I provide some unwanted comments or some, light comic relief, but occasionally an idea, and they like it and they work on it, and it comes to something. So I think that's a really important message, and the AGITG is kind of grease the wheel of that, because of the samples, because of the questions we can ask. And probably what we need to do more, we've tried but not as much as we could, is to get scientists involved in the design of the trials from the beginning. They need to sit with us when we're actually sitting there and writing it up. And I have some colleagues that I work with who keep hammering this home to me. And in fact, we're about to do a phase 1 immune trial asking a different immunology question. And one of my laboratory colleagues was involved from the beginning in the design of that trial and what the laboratory questions should be and what tissues we should collect as part of that study. So I think that's something that's important to celebrate is that we have this connection that Lorraine was talking about. It's a small group, but they do collaborate well together. I think that's important. The other thing I wanted to exemplify or amplify of Lorraine's is, it's true that the genetic revolution hasn't delivered as much as we hoped so far, but it's certainly provided a huge treasure trove of possibilities of what to try, so that's important. But heading backwards a bit and looking for commonalities instead of subdividing is perhaps going to be the way forward. And the APGI has even tried to form groups of mutations that logically sit together, right? 4 groups instead of 20, and seeing whether those groups of aggregation might still be susceptible to some types of treatments rather than others. But I think the Palbo and some others are interesting because they cut across different genetic mutations. So you can have 5 different types of mutations, but they might all be sensitive to this particular block in the message that comes from the cell surface to the nucleus, irrespective of the other mutations that are going on. And that gives us some pause for hope as well.